Hi all, welcome to Physio Exam Buddy. In this video, we are going to study a frequently asked short essay question, that is genesis of resting membrane potential. Genesis resting membrane potential. It is also known as transmembrane potential or resting membrane potential. Next is about the definition of the resting membrane potential. That is, it is the potential difference between the extracellular fluid as well as intracellular fluid along the cell membrane when the tissue is at rest and inside being negative relative to the outside. So that is the definition of the resting membrane potential, and it is usually denoted as negative. Regarding the recording of resting membrane potential, it is recorded with the help of a cathode ray oscilloscope. What you are doing for the measurement of resting membrane potential is that you will keep one electrode in the interior of an axon and another electrode will be placed on the exterior surface of a cell, and this potential difference between the interior as well as the exterior electrode is measured. That will give you the resting membrane potential, which is usually measured using the help of a cathode ray oscilloscope. The magnitude of the resting membrane potential, it ranges from -9 to -100 millivolt. In case of neurons, the resting membrane potential is usually -70 millivolt, whereas in case of skeletal muscle as well as cardiac muscle, it is around -90 millivolt. And the smooth muscle got no definite value for the resting membrane potential. It usually have a fluctuating resting membrane potential. Now we are going into the answer proper of the genesis of resting membrane potential. The question can also be asked as ionic basis of resting membrane potential. So how the resting membrane potential originate? It is basically due to the unequal distribution of the ions across the cell membrane. You all know that the major ions or the most abundant ions in the extracellular fluid is the sodium as well as chloride. That is ECF. It behaves like a seawater. That is, seawater usually contains sodium and chloride. So the ECF usually have the most abundant ion sodium as well as chloride. Whereas the most abundant ion in the intracellular fluid is potassium ions. So that is the basic thing you should know before going into the genesis of resting membrane potential. And when a question is asked like genesis of resting membrane potential, you have to explain the answer under these headings. First of all, you have to explain the selective permeability of the cell membrane. Second, about the Gibbs-Donan equilibrium. Third, the Nernst equation. Fourth, Goldman-Hodgkin-Katz equation. And fifth, the sodium-potassium ATPase pump. Now we will see in detail about each of these factors. First of all, the selective permeability of the cell membrane. You all know that our cell membrane in all parts of our body, it is selectively permeable. That is, it is freely permeable to certain ions like potassium as well as chloride, whereas it have moderate permeability to ions like sodium, and it is usually impermeable to the intracellular proteins. And size also depending on the size also the permeability differs. That is, the hydrated potassium, it is comparatively smaller in size when compared to that of the hydrated sodium. So the size difference also causes difference in the permeability in across the cell membrane. So first of all, we are assuming that this is the inside of the cell and this contains the ICF, and this portion, this white portion is the outside of the cell which contain the extracellular fluid. And you know the most abundant ion in the ECF is sodium and chloride, whereas the most abundant ion in the intracellular portion, it is potassium. So we are assuming that in this instant the membrane, it is permeable only to the potassium ions but not to any other ions. So you know that potassium, it is the major component of the intracellular portion. So the concentration of the potassium is more inside the cell. So this large potassium concentration gradient occurs from inside to the outside, which will pump the potassium from inside to outside. So along with this potassium, it will also carry a positive charge from inside to outside because the charge of the potassium is positive. So what happens? It will create a electropositivity outside the membrane, because now the potassium is more on the outside, which will carry the +1 charge along with the potassium. So it will create a electropositivity outside the cell membrane. And so what happens to the inside of the membrane? As positive charge carrying potassium is pumped out, the inside will automatically becomes negative. So there will be an electropositivity outside the membrane and an electronegativity inside the membrane. And there occurs a potential difference between the inside and outside, which is known as the diffusion potential. So in case of potassium, this potential difference is the -90 millivolt. And once this diffusion potential is attained, it will block further diffusion of potassium. So like that, the same thing which happens in case of sodium also. So at this instant, you just assume that this cell membrane is permeable only to the sodium ions. And you know, sodium is abundantly found in the extracellular fluid. So the concentration, the direction of the pumping will be from outside to inside. So the sodium will be also pumped from outside to inside. So there will be electropositivity inside the cell membrane when compared to the outside, which is becomes electronegative. And this pumping will occur till the diffusion potential of sodium is attained, and the diffusion potential of the sodium, it is found to be +60 millivolt. So from these two instances, it is clear that the concentration difference of the ions across a selectively permeable membrane under appropriate conditions, it will create a membrane potential. So one of the most important factor that helps in the genesis of resting membrane potential, it is the selective permeability of the cell membrane. Next factor which causes the genesis of resting membrane potential is the Gibbs-Donan equilibrium. The Gibbs-Donan equilibrium states that in the presence of a non-diffusible ion, the distribution of the other ions to which the membrane is permeable is affected in a predictable way. So please consider this M as the cell membrane and this X is the intracellular portion, because it contains the intracellular proteins which is usually impermeable through this cell membrane. And this portion, it is the extracellular portion. Okay, so M is a membrane which is impermeable to proteins, and there are two compartments, that is X compartment as well as Y compartment. Now assume that the initial concentration of the cations as well as anions, they are equal on both sides of this membrane. Now you know that chlorine, it is more found in the extracellular portion. So chloride will diffuse down its concentration gradient from the Y to X. And you know the charge of the chloride, it is negative. So along with the chloride, some potassium which is having an opposite charge also will move along with this chloride into the X compartment. So what happens to the concentration of the potassium in the X compartment? It will carry some potassium from Y to X. So the concentration of the potassium in the X compartment will be more when compared to that of the concentration in the Y. So in short, there exists an equation, that is concentration of potassium in the X as well as chloride in the X as well as protein in the X compartment, it is usually more than that of that in the Y compartment, because of the pumping of this chloride ions into the X compartment from the Y compartment. So Donan and Gibbons show that in the presence of a non-diffusible ion, so in this case the non-diffusible ion is the intracellular protein. The diffusible ions, that is the potassium as well as chloride, it will distribute themselves so that equilibrium their concentration ratios are equal. That is, at equilibrium the concentration of potassium in X by concentration of potassium in Y, this will be equal to the concentration of chloride in Y by concentration of chloride in X. By redistributing in this equation, you will get an equation like this, that is concentration of potassium in X into concentration of chloride in X is equal to the potassium concentration in Y multiplied by the chloride concentration in Y. Next factor affecting the genesis of resting membrane potential, that is the Nernst equation. So Gibbs-Donan equilibrium results in a concentration gradient across the cell membrane. So chloride ions are now present in higher concentration in the ECF that you all know. So they will diffuse along this concentration gradient into the cell. So interior of the cell becomes negative because chloride is having a negative charge. So the chloride will be pumping from the ECF to the ICF. So it will carry the negative charge from the outside to the inside. So interior of the cell becomes negative and the chloride will be pushed out of the cell along this electrical gradient. And an equilibrium will be reached then between both the concentration gradient as well as the electric gradient, creating a potential difference across the cell membrane, which is known as the diffusion or the equilibrium potential across the cell membrane. And the magnitude of this diffusion or equilibrium potential across the cell membrane, it is determined by the Nernst equation. And you should know the Nernst equation, that is the electromotive force in millivolts, that is the diffusion or the equilibrium potential. It is calculated by plus or minus 61 divided by Z, where Z is the charge of the ion into log of the concentration of that ion inside divided by the concentration of that ion outside. And the sign will be positive if the ion is diffusing from inside to outside, is a negative ion, and the sign will be negative if the ion is a positive ion. And Nernst calculated the equilibrium potential of almost all the ions across the cell membrane that prevent the net diffusion of the ions to inside as well as outside. So what Nernst have calculated as the diffusion or the equilibrium potential of the potassium is that Nernst actually made a hypothesis, that is ECF and ICF contained only potassium. That is, he is going to consider that only ion which is present in the ECF as well as ICF is potassium, and the cell membrane is actually freely permeable to the potassium ions. Now the potassium, you all know that it is more present in the inside of the cell. So potassium will diffuse down its concentration gradient through potassium leak channels from the inside of the cell to the outside of the cell. And along with the potassium, it will carry the positive charges to the outside. So there will be an increase in the negativity on the inner side of the membrane. This will oppose the negative electrical potential, which will tend to prevent the further exit of the potassium, positive potassium ions. So when this electrical gradient will be equal to the concentration gradient of the potassium, there won't be no net potassium movement across the cell membrane, and the potential at which this is attained, it is known as the Nernst potential or equilibrium potential or diffusion potential of potassium, and it is found to be minus 90 millivolt. Now we are going to explain the equilibrium potential of the sodium. You know that sodium, it is most abundant ion in the extracellular fluid. So again here, for explaining the equilibrium potential of the sodium, Nernst again made a hypothesis, that is ECF and ICF contain only sodium ions and the membrane was freely permeable to sodium ions. So the sodium will diffuse down its concentration gradient from the outside of the cell to the inside of the cell, carrying along with it the positive charge, because sodium is a positive charge ion. So more positive charge will be accumulating on the inside of the membrane, which will decrease the negativity on the inner side of the membrane. As this goes on, the positive charges will build inside, which will oppose the electrical potential and prevent the positive sodium from further entering into the cell. So again, when the electrical gradient will be equal to that of the concentration gradient, there won't be any net movement of the sodium across the cell membrane, and that potential at which this electrical gradient becomes equal to the concentration gradient, it is known as the Nernst potential or equilibrium potential of sodium or diffusion potential of the sodium, and it is found to be plus 60 millivolt. So here, like the sodium as well as potassium, the chloride has also got an equilibrium potential and it is found to be minus 70 millivolt. Next factor causing the genesis of the resting membrane potential is Goldman-Hodgkin's CATS equation, and it is used to calculate the diffusion potential when the membrane is permeable to several different ions. Please keep in mind Nernst equation is used when the membrane is permeable to only a single ion, but Goldman-Hodgkin equation is applied when the membrane is permeable to different ions. And what is this Goldman-Hodgkin equation? This diffusion potential, that is the electromotive force in millivolt, it will be equal to minus 61 into log of C means here concentration and P is the permeability. I is inside concentration and O is the outside concentration. So if you substitute this in this formula, you will be able to calculate the diffusion potential across the cell membrane if the membrane is permeable to multiple ions. And the last factor which causes the genesis of rusting membrane potential is the electrogenic pump, that is the sodium potassium ATPase pump. And you know that the main function of the sodium potassium pump is that it will pump 3 sodium ions out in exchange of the 2 potassium to inside, and this will contribute to the minus 4 millivolt of the rusting membrane potential. And some sodium also leak to inside of the cell through the leak channels in the rusting state, and this excess sodium is also pumped out by the sodium potassium ATPase pump, and the RMP is made constant with the help of this sodium potassium ATPase pump. So while explaining the genesis of rusting membrane potential, you have to explain all these 5 factors under these headings. Thank you.